Well, we still have some people trickling in. I welcome everyone to today's seminar, and it's really my great pleasure to welcome René Benas, who's joining us from the Netherlands Cancer Institute in Amsterdam. And while I'm sure he doesn't really need an introduction for most people here, let me still spend a minute or so and give you some background on him. René is a professor of molecular carcinogenesis at the Netherlands Cancer Institute in Amsterdam. And I've really known him as a true pioneer of using functional genomics and genetic approaches to identify drug combinations. And some of his early work has led to important clinical breakthroughs, such as the combination of BRAF and EGFR inhibitors in BRAF mutant colon cancer. And he's also been active in developing prognostic gene expression tests and other biomarkers. And this work has continued, and I'm not going to tell you much more because I'm sure he will. He has been honored for his work with many prestigious prizes and awards, including most recently the Ernst Bertner Award for Cancer Research and the ESMO Lifetime Achievement Award. He is also a member of the Royal Netherlands Academy of Sciences, the National Academy of Sciences, the American Academy of Arts and Sciences, and a fellow of the AACR Academy. Before I hand the floor to him, let me just remind you of two housekeeping items. So as you see on the screen, all of these seminars are recorded and will be available on the web. And also future seminars you can find on the web so you can plan ahead. And for questions, please use the Q&A tool. Don't post it to the chat and we'll read them at the end. So with no further ado, René, please go ahead. The floor is yours.
Thank you so much, Eric, for the kind introduction. I hope everyone can hear me and see my slides. I'm going to talk about rational drug combinations for cancer. This concept, of course, is not new at all because I'll show you some disclosures first. I'm working with two companies called Lixti and Heparigenics, and I'll talk about two of their drugs in preclinical experimentation today. So this whole concept of drug combinations is not new at all. The first paper that I could find dates back to 1958, combining methotrexate and 6-mercaptopurine for treatment of leukemia. So the answer to cancer drug resistance is easy. In principle, it's combinations, but it's hard in practice because we can make so many combinations of all the drugs that we have available. And the answer is probably that every cancer needs a different combination depending on the genotype and the tumor microenvironment. So it is not a simple problem, but we can begin to learn some of the basic rules. And we have been trying to do this through what we call functional genomic approaches, where we use genome scale gene inactivation tools to ask which are the most powerful drug combinations. And I want to talk today mostly about unpublished work, which has the drawback that some of the questions that you may have, I have no answers to yet. But the advantage, of course, is that you learn hopefully something new. And I hope that some of the unpublished work that I'm going to talk about will stimulate your thinking about some of the problems that we have in oncology. But I need to introduce our field with a few published examples. And I'll start with the example that I became aware of around 2011. This was the time that we had just seen the first spectacular results by Keith Flaherty and others that BRAF mutant melanoma responds very well to selective BRAF inhibitors. But at the same time, we learned that BRAF mutant colon cancers did not respond to BRAF inhibitors. So here you had a situation where the genotype is similar in terms of the BRAF mutation. The drug is the same, but the responses are very different. So we started asking, what else should we inhibit in the BRAF mutant colon cancer to make the BRAF inhibitor effective? So I won't dwell on these experiments. It was a short hairpin RNA screen that we did to ask which kinase should we inhibit together with BRAF to make for a response in these BRAF mutant colorectal cancer. And the answer that came from that screen was that EGFR co-inhibition was synthetic lethal with BRAF inhibition. And that was validated very quickly thereafter in a proof of concept clinical trial in BRAF mutant colorectal cancer patients. And that was followed up by a registration trial that was published in 2019. And that led to FDA and EMA approval for this particular combination in BRAF mutant colorectal cancer. So here you see the sort of nice linear path from discovery to clinical acceptance of such a therapy, which by some standards is relatively short, but I still feel that this is way too long and that we should be able to do this faster than 8 years in the future. So one more published study before we go to new work, because I also want to point out that you can apply the same technology to drugs that are already FDA approved. In this case, the example of a multi-kinase inhibitor that is used for liver cancer treatment, lenvatinib. It's a broad spectrum kinase inhibitor that delivers some clinical benefit, some 7-month progression-free survival benefit, and some 1 in 5 patients have benefit from this drug. So we thought there was significant room for improvement, both in terms of number of patients responding and duration of response. So we basically repeated the trick that I just showed to you. We looked for liver cancer cells that did not respond to lenvatinib, and that was in fact the majority of the cell lines in our panel did not respond to lenvatinib, just like the majority of patients don't respond to lenvatinib. And now, of course, we've entered the CRISPR era, so now we did a kinome CRISPR screen, very similar to what we did before with hairpins, in a BRAF mutant colorectal cancer cell that does not respond to lenvatinib and ask which kinase should we inhibit in conjunction with lenvatinib to get lethality. And the answer was once again EGFR. And very compellingly, nearly all of the guide RNAs for EGFR were significantly depleted in this experiment. So that left no room for interpretation. And indeed, we found that liver cancer cells that express significant levels of EGFR are very sensitive to the combination of lenvatinib and gefitinib as a small molecule inhibitor, or erlotinib as a similar small molecule inhibitor. But the cells that did not express EGFR obviously did not derive benefit. And that was also seen in xenografts, and I won't show it here, also in PDX models. So that set the stage for another clinical trial that for more practical reasons we performed in Shanghai, where the incidence of liver cancer is much higher than here in Amsterdam due to the endemic nature of hepatitis B and C virus in China. So working with a professor at Saibo at the Renji Hospital, we selected the patient cohort that were EGFR positive based on immunohistochemistry for EGFR, and started treating these patients post-progression on lenvatinib monotherapy by adding gefitinib to the treatment regimen. So first we allowed patients to undergo regular treatment, which is in China lenvatinib, standard of care. And then when they progressed on a scan, we added gefitinib and we asked whether that led to a response. And to make a long story short, we saw that out of 12 patients, 4 had stable disease, 4 had partial remissions, and 4 progressed. And that became a publication last year, which is now being followed up by the pharmaceutical industry with a large 400-patient trial randomizing patients to gefitinib versus or lenvatinib versus lenvatinib plus gefitinib. And we hope to start that trial by the middle of this year, again in China, for the reasons of patient availability. So now I want to turn to a more difficult problem and more relevant, I think, to large cancer populations, because KRAS is obviously mutated in a large fraction of human cancers, but the MEK inhibitors that are a logical candidate downstream of RAS delivered very modest clinical survival benefit in lung cancer and in other cancers. So the question is really, why is MEK inhibition downstream of RAS not sufficient? And we already tried to address this question back in 2014 when we did another of these synthetic lethality screens and found as a top hit ERB3, the member of the HER2 family. And that turned out to be a synthetic lethal hit because inhibition of MEK in both lung and colorectal cancer leads to a feedback reactivation of ERB3 that then superactivates the MAP kinase pathway to the extent that MEK inhibition is insufficient to block the pathway completely. That worked really well in cell culture. We see here a pan-HER inhibitor because ERB3 can form heterodimers with all the members of the ERB family. So you need to use what we call a pan-HER inhibitor like dacometinib. It showed elegant synergy in cell culture with selumetinib as a MEK inhibitor. It also worked beautifully in animal models, as you see here in the xenograft experiment, single drug and versus combination treated. So then we moved enthusiastically into the clinic and in fact did 3 different clinical trials comparing 3 different MEK inhibitors with various pan-HER inhibitors. So the PD901 as a MEK inhibitor with dacometinib, we used trametinib lapatinib as a pair, and afatinib selumetinib as a pair. And we tested it in KRAS mutated colon and lung cancer and some pancreatic cancers. And the answers were very disappointing, not because the combination doesn't work, probably, but most likely because the combination of MEK and a pan-HER inhibitor quickly leads to dose-limiting toxicities in patients. And that's because the toxicities of these drugs in patients overlap. MEK inhibitors and pan-HER inhibitors both yield GI toxicity. And as a result of that, you reach a dose-limiting GI toxicity before you reach an effective dose. This was not seen in the mice, which again reminds us of the notion that toxicity in mice doesn't really foretell toxicity in patients. So this was actually a bust, clinically speaking. So we need to reconsider. And one way to reconsider was a finding that we made back in 2018, an unexpected finding that tumors that have lost a kinase called MAP2K4 become exquisitely sensitive to MEK and ERK inhibitors. And you see here one seminal experiment from this paper in Cell Research from 2018, where we CRISPRed knockouts of MAP2K4 in quite a few different cell types. I show you here one, a lung cancer cell, and show that the knockouts are exquisitely sensitive to trametinib or the sharing ERK inhibitor. We have studied in detail how this works. I won't bother you with all the experiments. It is all published. But the notion is that receptor tyrosine kinases signal, of course, through the conventional MAP kinase pathway, but also through a pathway that involves MAP3K1, MAP2K4, JNK, and JUN. And these are 2 parallel pathways. And what happens when we inhibit MEK with a MEK inhibitor like trametinib, that leads to initial inhibition downstream. And it's important to point out here that MEK signals to ERK to activate a dual specific phosphatase called DUSP4. And DUSP4 is an inhibitor of the JNK kinase. And as a result of that, we see that the moment we inhibit MEK and ERK, we lose this DUSP phosphatase, and now the JNK kinase is activated. And now we see signaling through this pathway, through JUN, and JUN is a transcription factor that activates quite a few receptor tyrosine kinases that now reactivates the MEK-ERK pathway. This also explains why we found ERB3 as a hit in that synthetic lethality screen, because ERB3 is one of the most activated genes by c-JUN in our analyses. So why is this then relevant to MAP2K4? Well, the moment you have a mutated MAP2K4, there's no upstream signal into JNK and JUN that leads to a loss of this reactivation pathway in the presence of trametinib. And now there's no reactivation. The MEK pathway is silenced and cell death occurs, as we saw. So you could say, well, that's nice, but that's mostly of academic interest because nobody has made a small molecule MAP2K4 inhibitor that we could potentially use in combination with MEK inhibitors. That was the case until recently, until my colleague Lars Zender, who used to be a postdoc with Scott Lowe at Sloan Kettering, did an in vivo RNAi screen where he found that MAP2K4, otherwise known as MKK4, is a key factor involved in liver regeneration. And his data showed that if you inhibit MAP2K4, you greatly accelerate liver regeneration. And since he's a liver doctor, he was interested in making drugs that would facilitate liver regeneration in chronic liver disease. So he got together with a chemist called Stefan Laufer to make a new selective MAP2K4 inhibitor based on vemurafenib, because vemurafenib, the BRAF inhibitor, had initially a slight off-target activity towards MAP2K4, which they optimized to be far more selective for MAP2K4 and loss of the activity against BRAF. So Heparigenics made this drug. It's in phase 1 for liver regeneration, but I contacted Lars and told him that I thought there was a much better application for his drug in oncology. And this compound that we for now call HRXB, the MAP2K4 inhibitor, we tested in simple drug combination schedules in various KRAS mutant cancer models. We combined with trametinib, and you see here one of these conventional matrices where you increase trametinib in one direction, you increase the MAP2K4 inhibitor in the other direction, and then you ask, is the inhibition of the two better than the inhibition of two alone? And you can quantify that by performing a Bliss synergy score analysis across the entire matrix. And when the color in this lower diagram is dark red, it means there is strong synergy between the two drugs. And you can already see that at 5 and 10 nanomolars, there's very little activity of trametinib, but in combination with 6 to 8 micromolars of HRXB, it becomes suddenly very potent. And you see that here as well in another KRAS mutant lung cancer, in a KRAS G13D mutant colorectal cancer. Very strong synergy, and interesting and important, this is over a large concentration range, which makes it probably very suitable for translational use. That's of course very more difficult if it works only in a narrow concentration range. And that leads to induction of apoptosis. As you see here in the cleaved caspase assay, we see that the combination is a strong inducer of apoptotic death, which we don't see with the single agent. So that's good. Moreover, we see that other inhibitors in the MAP kinase pathway, such as the more recent KRAS G12C selective inhibitors, we use here AMG 510, sotorasib. This is a good inhibitor of KRAS G12C, specifically in lung cancer, but nevertheless, we can see that in low concentrations, we can reach dramatic synergy again by combining the AMG 510 with the Heparigenics MAP2K4 inhibitor. Again, shown in the synergy score. And also, again, you see the induction of apoptotic cell death that is resulting from the combination, but not so much from the monotherapies. Also here, another KRAS G12C lung cancer model. So, which other synergies do we see? We see synthetic lethality with SHP2 inhibitors. Here we use RMC-4550, the Revolution Medicine compound. Of course, obviously, this also inhibits MAP kinase signaling and again shows synergy with the HRXB compound. So all of these experiments are currently undergoing animal validation, and you could argue that I should only talk about it when it works in animals, but I want to show you where we are, because I think the clear conclusion is not only that rational drug combinations that we identify through genetic screens are more likely to give us durable responses in the clinic than many of the trial and error approaches that we see tested in the clinic today. But the other important conclusion is that I think that MAP2K4 is a new kid on the block, for targeting in combination with MAPK inhibitors in MAPK activated cancers. And we now have for the first time a tool to anticipate the first question. We see synergy in the range of 5 to 6 micromolars of this drug. And you could argue that that's a very high concentration of drug to use. But that, of course, all depends on how toxic this compound is in patients and in mice. And for the liver regeneration experiments, they can dose the drug very highly in human patients without toxicity. And in animals, we have reached easily 20 to 50 micromolar in plasma without any signs of toxicity. So yes, it's a low potency compound, but we also believe that it is not a toxic compound at the concentrations that we need to reach to make this work. So I now want to switch gears and talk to our activities that are based on what we now call the 1-2 punch model for cancer therapy. And we came to this model based on the 3 epic failures that I basically mentioned on the combination of MEK and pan-HER inhibitors that turned out to be too toxic to give to patients. So we thought, is there any way in which we can avoid combination toxicity? And we figured that that would be if we were able to get drug synergy without giving the drug simultaneously to the patient. And that can only work in a scheme where the first drug causes a new vulnerability that the cancer cell didn't have. And that vulnerability persists if you withdraw the first drug, and that you then come with a second drug that selectively kills the cell that has the acquired vulnerability induced by the first drug. This sequential treatment regimen would be synthetic lethality 2.0 because it's combination synergy while avoiding combination toxicity. But how do we make that work? The first one is, of course, are there any metastable states that are associated with novel vulnerabilities that also persist while we withdraw the first drug? And we figured that senescence was exactly what we need, because senescence is a stress response characterized by a proliferation arrest, which is good. The cancer cells stop growing. It is also characterized by a stable proliferation arrest that is not readily reversible, which is the first criterion to make the 1-2 punch work. And then another very attractive feature of senescent cells is that they produce quite a few cytokines that leads to an immune infiltrate that can help in clearing the senescent cancer cells. Another attractive feature of senescent cancer cells is that their transcriptome is very different, their metabolomic program is very different. So it might be possible to find vulnerabilities that are unique to senescent cancer cells that we can use to selectively kill them if the cytokine production doesn't lead to immune clearance by itself. So there's 2 questions here. How do we make cancer cells senescent and how do we kill them? And we have, in fact, then 2 tasks ahead of us: find senescence inducers, find senolytic drugs, a drug that kills senescent cancer cells, and potentially we could find drugs that modulate the cytokine profile such that the immune cells become more effective in clearing the senescent cells. That would be a 3rd possibility that I won't talk about today. I'll talk about the first and the second punch. How do we induce senescence? How do we kill senescent cancer cells? And does that work for us? So how do we do that? We do that with a reporter that was developed by Steve Ellich a couple of years ago. He showed that miR-146A was heavily induced in senescent fibroblasts, and he linked that promoter for miR-146A to GFP. And this vector, when expressed in proliferating cancer cells, is not turning on GFP. But when we then come back with a CRISPR library, then we can find genes whose inactivation provokes a senescence response in the cancer cells that turns on the GFP marker. So a small subset of the cancer cells will become GFP positives. These cells can be sorted out by FACS, and then we can recover the guide RNAs in these cells. And those are then the guide RNAs for the genes whose inactivation induces a senescence response. This works very well. And we published this already a couple of years ago, that we can do both compound screens and genetic screens to identify senescence-inducing agents. One of the most potent agents that we found are the aurora kinase inhibitors like alisertib and barasertib, very potent inducers of senescence. You see here the blue color is senescence-associated beta-galactosidase, a marker of senescent cells, but you also see that transcriptomic profiles of senescent cells are enriched in these cells. Markers like P21, DNA damage, are induced loss of phospho-Rb. These cells have all the hallmarks of senescent cells. So we can readily induce senescence as a stress response in cancer cells, but the tall order is, how do we kill them? What is the vulnerability of senescent cancer cells to make them die? And the reason we want to make them die is that long-term persistence of senescent cells can lead to a chronic inflammatory response. And we all know that chronic inflammation can lead to tumor progression, tumor dissemination, cell migration, and that's not a good thing to have persist in a tumor. So we now have a neon sign in my lab that says the only good cancer cell is a dead cancer cell. And that's because we worry that persistent cancer cells may, of course, re-emerge and repopulate a tumor. So we would rather kill this cancer cell than let it sit around and provoke a chronic inflammatory response. That's why we need senolytic drugs. So how do we do that? This is the work of Liqing Wang in my lab. He designed a clever CRISPR screening library that was a doxycycline-inducible CRISPR library that we introduce in proliferating cells first. Then we induce senescence with a drug like alisertib or barasertib. And then only after the cells have fully become senescent, we activate the gene editing by adding doxycycline in the non-dividing cells. And we ask which guide RNAs can mediate cell killing in the non-proliferating population. So we compare the non-dividing senescent cells to the senescent cells that have been incubated for 2 or 3 weeks with the gene knockout to see what is specifically depleted. We, of course, also compare the proliferating arm P2 versus P1. These are simply the essential genes that are required for proliferation. Those we don't want. So we filter out P2 versus P1 differences and only retain selective S2 minus S1 differences. We did these screens with multiple senescence inducers, PLK1 inhibitors, PLK4 inhibitors, aurora kinase, chemotherapy-inducing agents. And if we look at what drops out, we find 2 genes repeatedly. We find BCL2L1, component of course, the intrinsic apoptosis signaling pathway, and C-flip, the regulator of extrinsic apoptosis pathway. So clearly indicating that apoptosis resistance is a hallmark of senescent cancer cells, and that perturbing either intrinsic or extrinsic apoptosis signaling is the way to selectively kill senescent cancer cells. So C-flip, as another name for C-flar, C-flip is the negative regulator of death receptor signaling. And death receptor signaling is mediated by the 2 death receptors, DR4, DR5, that bind their ligand called TRAIL. And when TRAIL binds the death receptors, it leads to caspase activation and apoptotic death, unless a high concentration of C-flip can inhibit death receptor signaling. And what you see here, that if we make CRISPR knockouts for C-flip, that leads to massive induction of cell death selectively in cells that were rendered senescent by either aurora kinase inhibitors, chemotherapy, or a PLK inhibitor, but not so much in parental cells. So we see highly selective tumor cell killing of senescent over proliferating cancer cells. And the reason that senescent cancer cells are sensitive to C-flip depletion is shown here. We see that alisertib or chemotherapy or PLK-induced senescence leads to an upregulation of death receptor 5. It leads to upregulation of TRAIL, and it leads to upregulation of C-flip. You see that also at the protein level. So we have a cell that is primed for apoptotic death because the ligand is up, the death receptor is up, but they don't die because the inhibitor C-flip is also up. So there's a dynamic equilibrium that on the one hand, DR5 signaling is up, the receptor and the ligand, but the inhibitor is also up. And now you understand why removing C-flip is such a powerful event, because now it unleashes the full activation of the death receptor mediated by increases in TRAIL and DR5. And this we see in virtually all cancer cells. Not only do we see it in all cancer cells, but it's also totally independent of how we make cancer cells senescent. So here you see a simple experiment where we use an antibody that activates death receptor 5 called conatumumab. And conatumumab is very inactive on proliferating lung cancer cells, A549, but is about 10-fold more sensitive in senescent A549 cells. And you see that here also for gamma radiation, an inducer of senescence, conatumumab as a DR5 agonistic antibody doesn't kill proliferating lung cancer cells, but kills gamma irradiated lung cancer cells. Now, the best senolytic drug that is currently described in the literature is a drug called navitoclax that is a BH3 mimetic that acts on the intrinsic apoptosis signaling pathway. And I showed you that indeed components of intrinsic apoptosis signaling were also hits in our screen that we performed. But there's quite a few cells that are insensitive to navitoclax. In fact, 6 of the 14 cancer cell lines that we tested did not respond to navitoclax when made senescent. But all of the cell lines that we test respond to conatumumab with a rather impressive ratio that we call the senolytic index, how much more sensitive are senescent cells over proliferating cells to this drug. And we see that on average, it is between 5 and 10-fold at least for the majority of cancer cell lines. So in vivo validation shows that indeed alisertib leads to tumor stasis as a senescence inducer here in a hepatocellular PDX model. Conatumumab as a single agent doesn't do a lot because it's a senolytic, it only acts on senescent cells. And you see that the combination, in fact, leads to tumor regressions. A very interesting phenomenon we see when we use barasertib as an aurora kinase A and B inhibitor, because here we see only very marginal tumor control by barasertib, the sort of orange growth curve in vivo. The conatumumab doesn't have much of an effect, but now we suddenly see complete tumor control by the combination. And that's weird because if conatumumab can only kill the senescent cells and only very few cells become senescent as judged by the barasertib monotherapy treatment, how is it possible that the combination suddenly leads to tumor control? And you see that also here, if we stain sections of barasertib treated cells for P21 as a senescence marker, you see that at best, maybe 20% of the cells are P21 positive, meaning that 80% of the cells are not senescent. Yet we get a very interesting tumor control if we now add conatumumab. You also see, of course, that all the senescent cells are now killed, but apparently we kill far more cells than only the senescent cells as judged by this tumor control. And that argues for a bystander effect meaning that conatumumab will now also kill in the presence of senescent cells quite a few non-senescent cells. And we tested that preclinically in cell culture, arguing that, of course, senescent cells produce paracrine factors. Of course, one of them is TRAIL that is secreted because I showed you how much more TRAIL senescent cells produce, but they also make cytokines like IL-6 and IL-8 that are known inducers of NF-kappa B in neighboring cells. And of course, NF-kappa B induces DR5 and induces TRAIL to get these cells sensitized to a DR5 agonistic antibody. So we think there's potentially a mechanistic rationale that paracrine factors secreted by senescent cells will kill non-senescent cells that are in close proximity. And I want to show you one experiment that we did to support that. If we take SASP conditioned medium, this is conditioned medium from senescent cells, and add it to proliferating cells, we see that these cells are sensitized to conatumumab killing, here comparing control medium to senescent conditioned medium. And that may provide an explanation for what we saw in vivo, that there is an enhanced killing of the cell of the tumor when we combine an agent that is only marginally effective in inducing senescence with the DR5 agonistic antibody. So I think we have evidence that there is a possibility to combine pro-senescence drug with senolytic drug in a sequential fashion, where we would get indeed combination synergy without combination toxicity. So I think we should reevaluate the importance of senescence induction in cancer therapy, because I think it provides us with new opportunities to treat cancer selectively. Now, in the last part of my talk, I want to show you something that I think is rather crazy, but I think is also at the same time potentially very, very interesting. So Mateus Enrique Diaz, my Brazilian postdoc, and I wrote an opinion piece or a perspective that we said, would it not be possible to consider activation of mitogenic signaling in cancer as a cancer therapy, as opposed to our ongoing efforts to inhibit oncogenic signaling? And this term paradoxical intervention is borrowed from psychotherapy, because I read a review on paradoxical intervention in psychotherapy, where it was described as a deliberate reinforcement of the pathological behavior to improve the clinical condition. And such a counterintuitive approach can be considered when more conventional interventions fail. So we thought the oncology equivalent of a paradoxical intervention in psychotherapy would be, instead of inhibiting oncogenic signaling, we would try to superactivate oncogenic signaling and see whether we can use that in a therapeutic setting. Now, if you think about this, we of course need to appreciate that oncogenic signaling, when it's hyperactivated, provokes a number of stress responses in cells, because we know that cancer cells are highly stressed. We typically see DNA damage stress, we see oxidative stress, mitotic stress, proteotoxic stress, and metabolic stresses. And that these stress response pathways are stretched to the maximum to accommodate the hyperactive oncogenic signaling. And that also at the same time means that a cancer cell will probably not strive for maximal activation of oncogenic signaling, but rather for an optimal oncogenic signaling. Oops, now I lose my screen here, just a sec. The cancer cells strive for optimal oncogenic signaling, because too much oncogenic signaling provokes hyper stressing of the stress response pathway and leads to cell death. So while conventional therapies aim at inhibiting oncogenic signaling, we asked what would happen if we superactivate oncogenic signaling and potentially combine that with a perturbation of the stress response pathways that the cancer cell employs to deal with oncogenic signaling. So this is a sort of combination strategy based on deliberate hyperactivation plus perturbation of stress responses. Does that work for us? So the first question is, how do we hyperactivate oncogenic signaling? And there we are helped by the notion that there are small molecule inhibitors of a protein phosphatase called protein phosphatase 2A, or for short PP2A. And PP2A plays a role in many biological pathways, but PP2A is also known to inhibit AKT, beta-catenin, GSK3 beta signaling. It is also known to inhibit MEK-ERK signaling. So imagine that if you were to inhibit PP2A, you would lose the inhibition of Wnt, AKT, and MEK signaling, leading to superactivation of that signaling. Of course, PP2A is known to play a role in numerous pathways, including many DNA damage response pathways as well. So there is a small molecule inhibitor of PP2A called LB100. And LB100 indeed provokes hyperactivated oncogenic signaling. This is an SW480 colorectal cancer cell line that already has a KRAS and an APC mutation. So Wnt, MAP kinase are active, but we can superactivate the MAP kinase and the AKT pathways by incubating with increasing concentrations of LB100. We also see that superactivation of these pathways indeed leads to superactivation of the stress response pathways. I show you here a few: DNA damage, gamma H2AX, phosphohistone H3 as a mitotic stress response, and IRE1 alpha as the unfolded protein response. And this was not only seen in one colon cancer cell line, it was seen in a whole spectrum of colon cancer cell lines. We tested 7 in total. And in all of them, there was a commonality of induction of mitogenic signaling and induction of various stress response pathways. So it is remarkable that, in fact, this PP2A inhibitor, LB100, is not highly toxic to colorectal cancer cells. If we do a dose titration curve, we can see that the cells actually also in long-term proliferation assays can tolerate up to 5 micromolar of this drug, and there's still considerable proliferation. And at 5 micromolar, there is considerable activation of mitogenic signaling and also considerable activation of the stress response pathways. What is also interesting that a phase 1 trial with LB100 showed that there were very modest toxicities of this compound. No grade 3 toxicities were observed in any of the patients, whereas there was clear evidence of massive PP2A inhibition as judged by an increase in protein phosphorylation in peripheral blood lymphocytes. So this is interesting that, contrary to popular belief, PP2A inhibition may not be all that toxic to individuals. So I showed you, or I told you, that I think that maybe these drugs that superactivate mitogenic signaling become more effective as anti-cancer agents when you at the same time block these these stress response pathways. And Matthäus made a collection of 164 drugs that each act in any of these DNA stress response pathways. So he had drugs that acted on metabolic stress, inhibited proteotoxic, mitotic, oxidative DNA damage stress, or apoptotic stress, and asked for each of these compounds, is there any compound that is more effective in cells treated with LB100 PP2A inhibitor versus control cells? And he then identified the area under the curve, the degree of curve shifting, where the LB100 curve shifted to the left, or the compound shifted the response curve to the left in the presence of LB100 compared to control. And that you see graphically represented here on the right, a dot below the 0 means that the drug made the cells more sensitive to LB100. And you see here indeed that there are drugs like navitoclax and a drug that targets BCL-XL as intrinsic apoptosis signaling inhibitors that are effective here. But you also see drugs that are acting on WEE1 and CHEK1, multiple drugs in the CHEK1, WEE1 pathway and PLK1 pathway, suggesting that mitotic stress is also a potential synthetic lethal event with activation of mitogenic signaling. Now, I want you to keep this in mind that we have here WEE1, CHEK1, and PLK1 as hits, because Matthias also did a CRISPR screen, genome-wide CRISPR screen, exactly on the same cell, treating with LB100, asking which gene, when inactivated, sensitizes cancer cells to LB100. And the results of that screen showed, again, here the hit WEE1, which we also saw as a hit in the chemical compound screen in the form of adavosertib, the AstraZeneca drug that is a WEE1 inhibitor. So we went on to validate how strongly synthetic lethal are LB100 and adavosertib as a WEE1 inhibitor in a panel of 7 colorectal cancer cell lines. And the synergies, as judged by this ZIP synergy score, are quite remarkable over a large concentration range again. If you sum them up, it is a remarkable synergy in 7 models. You can also see that in short-term cultures, where the purple line is the combination of the 2 drugs and show you much better growth control than the single agents or the controls. And that leads to, again, these combination graphs where we show that the single agents are far less effective than the combination treatments in a whole panel of cell lines. So this looks very attractive from a potential treatment strategy. On the one hand, we activate mitogenic signaling, and then we inhibit the WEE1 kinase, and WEE1 kinase, of course, prevents mitotic entry prematurely. So now we push the cells into mitosis by inhibiting WEE1. And that's exactly what we see happening if we do time-lapse filming, which we did with Anouk and Jonne from the René Medema lab. We saw that cells that are treated with LB100 have massive DNA replication stress from the hyperactivated oncogenic signaling. And when you then push these cells into mitosis by treating with WEE1 inhibitor, we see that there is a mitotic catastrophe. The cells enter mitosis without having replicated their DNA, and now they crash in mitosis. So this looks like a very interesting model for cancer therapy. But why are we so excited about this? The reason that we are very excited about this as a treatment strategy, and again, this is very preliminary work, but I still want to share this with you, that we actually also started asking, how does a cancer cell develop resistance to LB100? How do you become resistant to superactivation of oncogenic signaling pathway? So we did a genome-wide CRISPR resistance screen now in a high concentration of LB100 to ask which genes, when inactivated, confer resistance to LB100. And when we did that, we saw beta-catenin, BCL9L, which is a component of Wnt signaling, but also components of MAP kinase signaling. And if you put all the hits in a string diagram, you see clearly 2 nodes. You see the beta-catenin node, and remember, these are beta-catenin activated tumors due to APC mutation, and they are KRAS mutant tumors, and we see this, this node of RAF MAPK1 that are hits in the screen. So clearly, it identifies downregulation of oncogenic signaling by loss of the oncogenic driver as a potential resistance mechanism to this treatment. And the only data that I have now to support this is that if we knock out beta-catenin in SW480 cells, you see that they become relatively resistant to LB100. So this makes sense. If you try to inhibit an oncogenic pathway, the cancer cells mutate to become resistant by reactivation of the pathway. But if we now do the inverse, we superactivate the pathway, the tumor cells try to mutate themselves to a lower level of oncogenic signaling, which they can accomplish by knocking out beta-catenin. And now they can tolerate higher concentrations of LB100. Of course, this is the oncogenic driver pathway that caused the cancer to begin with. So we argue that if you mediate activation of oncogenic signaling as a treatment strategy, you could potentially get tumor suppressive resistance to therapy, which would mean that if you now withdraw the therapy, that the cells will grow much less aggressively because they have lost an oncogenic driver mutation. All of these validation experiments in animals are ongoing, of course. I don't have data to show you yet. We are 2 weeks into treatment, so it's early days, but we'll have data to show you shortly. One thing that we can say is that LB100 and adavosertib is not very toxic in a combination, so we are optimistic that we can make this work in animals. So my last take-home message is that maybe we should also consider hyperactivation of oncogenic pathways as a treatment for strategy for cancer, because it could lead to very interesting and appealing resistance mechanisms. So I would like to stop by sharing some of the people's names that were involved in this work. The work on lenvatinib was done by Hao Jie-jin, who is now back in Shanghai. The paradoxical intervention that I mentioned last was done by Mateus Henrique Dias and João Neto. Senescence induction, the DR5 agonistic work was done by Liqing Wang and Hao Jie. The MAP2K4 work that I showed was done primarily by Robin Janssen in collaboration with Heparigenics. These are the people that fund our work, and I'll stop here. I thank you for your kind attention and will be happy to answer questions.
Thank you. This was great. We already have a couple of questions, but maybe I start with one. Your results on the priming effect in the case of a few senescent cells priming the entire tumor to sensitivity, does that suggest that probably it would be even more informative to do some of these screens in vivo?
Yes, potentially. You know, in vivo, CRISPR screens are not easy to do. You often lose complexity. Not all tumor cells are equally good at populating a tumor because of the presumed cancer stem cell effect, where only a minority of the cells grow out. So we have seen cells where we lose complexity in no time, but we have also seen cells where we don't lose complexity. So I don't think you can make a general statement whether it's possible or not. I think it depends on the cell type, how polyclonally they grow into tumors in vivo. So if you have such a tumor, it could be done, and then you could potentially do this. Yes, absolutely.
Okay. Let me get to some questions. There's a pretty good number already. I'll start out with a question by Jinjing Liu. Any blood-brain barrier penetration information for the new MAP2K4 inhibitor?
Oh, it's very early days. Of course, the company never bothered much about the MAP2K4 inhibitor blood-brain barrier because they designed it originally for liver regeneration and not so much for brain tumor treatment. So we don't have information on this yet.
Okay. Next question is by Lee Ellis. Is your induction of tumor cell senescence p53 dependent in any models? Are your combinations inducing immune cell, NK, and/or T cell infiltration and tumor killing?
Yeah, that's a good question. Sometimes the senescence is p53 dependent, but clearly in the case of alisertib, barasertib, this happens in every cell, cancer cell, irrespective of p53 status. We have previously published that CDC7 is a senescence inducer, but that happens only in p53 mutant cells, because then they cannot control their replication anymore. If you stall replication with CDC7 inhibition, a normal cell would mount a p53 dependent replication arrest, but the p53 mutant cells have no arrest left and mount a senescence response. So in some cells, we see that p53 is required for the senescence response. In some cases, the absence is essential to provoke the senescence response. And in some cases, it really doesn't matter. When it comes to the immune infiltrate, they're highly dependent on the cell type that you make senescence, not so much on the senescence trigger, but the cell type that becomes senescent dictates what the cytokine profile will be in the senescent state. And that makes that the cytokine profile that is produced and the resulting immune infiltrate is somewhat difficult to predict upfront. We do see a lot of immune infiltrates, sometimes more NK cells, sometimes more T cells, sometimes more macrophages, but there is always an immune infiltrate. Sometimes, and Scott Lowe was the big person to show first that there can be PD-1 synergy with senescence-inducing drugs, because you can activate the CD8 positive T cells with PD-1 that infiltrate the tumor. But we have also seen models that show no synergy with PD-1, apparently because the T cells are not massively attracted to that tumor by the SASP of the senescent cells. So it's somewhat poorly understood at this point how the SASP shapes the immune infiltrate and what are the rules. Can you predict it up front? At this point, we cannot.
Next question is from Keith Loeb. Does the strategy to target senescent cells also target tumor dormant and tumor stem cells?
That's a very good question. Whether dormant cells are targeted, because we think that dormant cells are a form of senescent cells. We are now focusing in my lab on what we call the drug-tolerant persister cells, which you could also call a form of tumor dormancy. We are performing the same type of CRISPR screens in drug-tolerant persisters as we did in senescent cells. And it's very early days, but we think that the vulnerabilities of drug-tolerant persisters and senescent cells are different, not necessarily overlapping. So I think that drug-tolerant persister cells, I don't dare to say whether all dormant cells have that same vulnerability, but drug-tolerant persisters seem to have different sensitivities to killing than senescent cancer cells.
Then we have a next question from Karin that goes in the same direction. Do you think by inducing senescent cells and subsequently targeting them, you're also targeting cancer stem cells?
Yeah, we don't know that. They are, of course, slowly proliferating. Whether they have the same sort of stress responses as rapidly proliferating cancer cells, I don't know. I simply have no data on that.
Yeah. Then we have another question by Keith Loeb. Does LB100 promote transformation in normal cells?
Yeah, the answer is that so far, it doesn't look like it. We can treat animals quite long-term with LB100 without seeing a lot of toxicity. And it is already in a phase 1b trial for a small cell lung cancer without, and some patients are already on treatment for quite a long time without any overt signs of neoplastic growth. That was my biggest concern, obviously. It doesn't seem to happen. And maybe the difference is, and I think the CRISPR resistance screen that I showed you maybe already begins to point at that. If you downregulate the oncogenic signaling, you're less sensitive to LB100, saying that normal cells that don't have hyperactive oncogenic signaling have not much toxicity from LB100, and they can buffer their mitogenic signaling much better than cancer cells can. So maybe this is not such a problem as I had feared originally when we started this project. But of course, time will tell. There is more and more patients being treated with LB100, so we'll find out soon whether this is a real issue or not.
This is a question by me. Do you know how potent LB100 is in comparison to things like ocadaic acid, which obviously also hits PP1, but just to get a sense where you are in the potency rate?
Yeah, I think that PP2A as a target in oncology got a bad name because of ocadaic acid, because ocadaic acid is so toxic. You cannot use ocadaic acid in vivo, to my knowledge, other than topical skin application, but not systemic. LB100 is a very different molecule because ocadaic acid is a natural product, and LB100 is a very small molecule, some 400 Daltons only. So they don't have any structural resemblance, and therefore it behaves very differently from ocadaic acid. So while I had predicted that PP2A would be very toxic inhibition based on the ocadaic acid, based on the multitude of pathways in which PP2A is involved, the data so far from the clinic say the opposite.
Yeah, I mean, oncogenic acid is a PP1 inhibitor as well, so it's much.
Yeah.
So right, I don't know if LB100 is explicitly selective for PP2A, and it might just be weak, which is sufficient to push the activation, but insufficient to cause real toxicity.
Yeah, I think it's an interesting molecule, and there's also very recent data from multiple labs that show that if you inhibit PP2A, yet you induce an MSI phenotype. And in preclinical models, there is strong synergy with checkpoint immunotherapy for that reason. So this may lead to another synergistic effect where LB100 not only hyperactivates mitogenic signaling, but also provokes an MSI phenotype by downregulating MLH1 and thereby creating neoantigens, making the tumors more antigenic. And that could be an additional benefit from this type of therapy.
There are more questions, but I think given the time, we have to stop here. Thanks, René. This was really great.
Thank you so much, Eric and others, for your time. And I look forward to meeting you sometime again in the near future.
Yeah, probably next week.
Maybe next week if you come to AACR. I'll be there. Thank you all.
Thank you. Bye.
Have a good day. Bye-bye.